We then talked about autophosphorylation. So if we come over here, we can see that the next part of the statement is about the autophosphorylation. Just get rid of this. Autophosphorylation of JAK1. So there's JAK1, and if we go zoom in here, we can see that it's now phosphorylated. Okay, it's got a P after here, which is the same as the one above, but the JAK1 is now phosphorylated. So we need to show that process happening, and of course this process being an autophosphorylation doesn't require the interaction of anything else, and so we can simply draw an edge from here to show that that process has occurred. Now if we read on, catalyze the phosphorylation, so once the activated complex catalyzes the phosphorylation of STAT2, so there's STAT2, okay, and we're going to phosphorylate STAT2, and it's this guy here that causes that phosphorylation. So there's STAT2 coming into the reaction, and there's the phosphorylated form of STAT2 coming out of that reaction. Now, in order to illustrate that it's actually this guy who's performing that phosphorylation reaction, again, we can make nice straight lines, are always good. We can change that edge, and again, clicking on an edge or a line allows us to change the arrow type here. And the catalytic thing is a purple reaction, and apply. So what we've now done is state that this guy here is phosphorylating STAT2. Okay. If we read on, STAT2 then binds to, let me just go over this way. STAT2 binds to STAT1, so there's STAT1 coming in. And now not a phosphorylation reaction, just get rid of that. It's a binding reaction. And that binds to there. And obviously what we get, we have to show the result of that reaction. And there we go here. Again, getting the lines nice and straight. That is the output of that. This then binds IRF9. IRF9 comes in here, and that's the product. So there's the binding event. And there we go here, out it comes on the other side. And then this translocates, so this is a cytoplasmic form of the transcription factor complex, and this will then translocate in the nucleus, and we can cause that there, translocating to the nucleus. And there we go. And then what we're going to show is that this guy here basically then activates and binds to the promoter of that gene, that gene, that gene, that gene, and that these are all going to be activated. Okay. And then in one case here, we say SOX3 inhibits the autophosphorylation of the receptor. So this is a feedback inhibition loop that is activated. So quite often when you're looking at receptor complexes and activation of systems, then some of the first proteins or genes which are activated are things which then will go back and block that reaction. So you're essentially having a feedback loop that feeds back and stops further activation of the system. So activating a system is quite easy. Stopping it and keeping it under control is often the more important thing. So we're going to now finish this off by saying that, oops, see daisy, well I'll just make that connector there first since I've started making a ledge. So let's draw that there. And let's draw a line from SOX3, over here. And just to make it neater, we'll draw it a bit like that, coming from here. And then we're going to draw an inhibitory edge, because what we're saying is that this inhibits that reaction. So let me just zoom in a little bit. So again, what we're going to do is change the nature of that edge to indicate that it's an inhibitory reaction. and we always use red to illustrate that, and we can do that there. So there we have this nice reaction. Let's make it nice and neat and make that line straight. So straight lines, even placing of components just helps the readability. Unfortunately, there is no automated layout algorithm that even if you had all of these relationships already defined, that when you could press a button and you'd get a diagram that is read. Now, in certain respects, the one thing that's missing from here is the idea that we actually have different compartments in the cell, and it's very important to us that we represent different compartments. So I'm just going to sort of draw a node here, and what I'm going to do is show you how you might want to represent a compartment. So nice properties. Let's take out the label here. Let's make it a nice, or I don't know, I'm just going to randomly choose a color here, but the principle is the same. We'll call this the cytoplasm. And we're not going to have a line color, and we're going to say, call it 300 by 300 for now. We'll apply that. And then we can say, so we imagine that these components are sitting on the cell surface membrane. We haven't drawn that membrane yet. We can do if we want. And then we're going to just drag that out behind it like that. And that's the translocation of the complex into the nucleus, so that's the boundary here. And so we've called this the cytoplasm, and we're just basically going to say, we're going to move that label so that it doesn't get in the way. So we're going to say internal, and we're going to call it the top, apply. And perhaps what we should also do is make it nice and big so it's quite obvious what it is that we're talking about, apply. And so there's the cytoplasm, and of course what we've got here, it's overlaid here. So we're going to lower the selection to the back. And so also what we have to do is now go back to that preferences and display. Okay. So we could draw a similar compartment for the nucleus. We could draw a similar compartment for the membrane. And essentially what we're doing now is not only drawing the relationships, but drawing the context in which those relationships takes place. So just to, I suppose, do almost a Blue Peter, as they say, we can open up this diagram here. So, okay, so that's the kind of ready-made version. So I'm hoping that if you look at that, you will see that as a reasonable representation of the text that you've been given, that shows now the extracellular membrane, the cell membrane, the cytoplasm, the idea that this all happens in the nucleus, and that there's this feedback loop there. So does that make sense? That kind of, so you could have all have done that with enough time, with a bit of training. It isn't difficult. It's just about understanding what the job is that you need to do, to understand a little bit about the tools and the way that you can do it and the kind of information that you want to convey. And also within this text, ignoring the bits of text here which are not relevant to what you actually want to represent within the diagram. And obviously this has been specifically designed where I've given you the exact names of the proteins as I want them to be represented, rather than the often loose use of component naming that is found in the literature. And obviously to say, even within a review article, things will rarely be described in such concise language as I've given you here. So this might be a whole paper that describes this process, and you'd still then have to untangle that information to pull out the information to give you this diagram. So I think this is a reasonable place to stop now, and for us to go and relax our brains for 15 minutes. But I hope that's proved one thing, that we can use this notation to describe a complicated process, that actually doing that is possible, but it isn't easy, because part of the problem is you've never done it before. But actually it's quite useful to be able to do it, because you end up with a diagram that summarizes a whole load of text in a nice, easy to digest way. And actually it's almost easier once you get your eye in to read this than it is to read that and build up a picture of what's happening. So what we've actually drawn here is a feedback loop. Yep. So how would that work, guys? If you think about this, what would be the dynamics of that system? And that's what we're going to explore after the break, where we're going to be able to say, okay, but how would that work? What are the assumptions that we're making in this diagram, and how that little feedback loop begin to actually regulate the genes down here, and this feedback loop work to control the system dynamics. Okay.